Light strengthens our tempo to maintain focus and clarity during no sudden changes or distracting me. Gentle and dancing with a soft piano notes. Light strengthens our tempo to maintain focus and clarity during no sudden changes or distracting me.
No, viewers at home, this is another opportunity for us to learn at Omega Bio-Edutainment as well as Omega Bioverse. Uh, in my previous videos, I want to believe that we were able to learn about the bacterial structure, a typical dividing bacterial structure, and in furtherance of that, that is why we are here once again. Don't forget that I told you then that I will be picking all these structures one after the other. We will now look at the structure, what are each of them made up. Uh, we will look at the function, we look at the physiology. We are going to see how each of these play a very vital part in their ecological role, that is, in the ecological role of such microorganisms, especially bacteria. Then we also look at a typical case study, giving us the example at which they have been able to maneuver this type of what nature has given them as a kind of a structure. Don't forget that I told us that the analogy is that each of these structure is like the various parts of human body. When you look at human body is composed of different parts that that have different functionality and perform different role. They have different structure, physiological role, ecological role, and we will also look at these examples in each case. Don't forget that in the previous class on the body plan of of bacterial cell, there are certainly about 9 to 10 basic parts of a dividing bacterial structure. The cell wall, capsule, that is number two, or what we call the glycocalyx. After that, we look at ribosomes, and after that, cell membrane is part of these very important parts. Plasmids play a very vital role as being part of a dividing bacterial cell. Pili, or what we also call fimbriae, we also play a very vital role. We are going to look at that. Then we'll also conclude it with mesosomes, nucleoid, cytoplasm, as well as flagella. So we are going to look at that. So I remain Adeotola Tunde Michael, the CEO of Omega Bio-Edutainment as well as Omega Bioverse. Just like I said, this afternoon we are going to look at defining bacterial cell, the body plan, and their composition. Okay, let's try and look at the general view of microorganism, the dividing cell. So just like I told you, this is the three-dimensional structure of a typical bacterial cell. This is a capsule, this is where ribosomes is being located, and here is where we have the cell wall, then as well as the pili is here. These are pili flagellum as well. Okay. Now this presentation is going to explore very important component of a dividing bacterial cell. We will then examine and explain their structure, that is their structural implication. Why are they there? So we'll look at the function as well as the ecological significance of each of these structure. Okay. Now the first we are going to look at is mesosome. The structure of mesosome are folded invagination, that is internal. They are folded imagination of the plasma membrane. That is when the plasma membrane, which happens to be this one, is infolded, is invaginated. So it's going to appear as a vesiculite or in form of a kind of a tubular projection, which is going to be inside the cytoplasm. Okay, so mesosome are very rich. Mesosome is a very rich structure that are enzyme rich and contain phospholipid, that is lipid that have phosphate group attached to them. Okay, there are the function of a mesosome. Number one, it is the site for cellular respiration and ATP generation. They perform that role. They help in DNA replication as well as they play a very vital role distribution um, during binary fission, as well as aiding information of cell wall by the anchoring enzyme. Don't forget that I told you that mesosome is an invaginated cell membrane. So, and it contains a lot of enzymes. So, all these enzymes are playing a very vital role in how the DNA is going to get replicated, as well as which contains a lot of enzymes. Okay, now the some of the functions also include they perform a very vital role during cell division. That is, you see them; they are very common. If it is not if the bacterial cell is not a dividing cell, you might find it very difficult to find them. And that is why what we are considering is a typical dividing bacterial cell, so that we'll be able to see some other ultra structure that are very, very important in the cell division. So what we are saying in essence is that mesosome play a very vital role in during cell division. Then it provides enzymatic services, which is very similar to what mitochondria also provide. So it is very, very important. An ecological role that they play is that they help in the survival of microorganisms even in low energy environment where energy is even in a very small supply. Because of the possession of mesosome, you find these microorganisms, you find them possessing mesosome. And examples of that is what we see in Bacillus subtilis, which uses mesosome to adapt to their environment during spore formation and when they are unnecessarily under stress. So the second structure we are going to consider is the nucleoid. The nucleoid, which is an irregularly shaped region within the cell which house or contain the bacterial circular membrane. Don't forget that I told us in the first video that we are dealing with prokaryotic organism, and prokaryotic organisms or prokaryotic cell is a kind of cell that doesn't have membrane bounded organelle. So all these organelles, you cannot even call them organelle because organelle are membrane bounded. So most of the structure within a bacterial cell, they are scattered, they are non-localized. You cannot actually pinpoint, for example, in eukaryotic organism or eukaryotic cell, you can actually decide where it's going to be, the nucleus is going to be located, where mitochondria is going to be located, where Golgi apparatus and endoplasmic reticulum, where they are going to be located very close to each other. Unlike what we have in eukaryotic cell, prokaryotic cell, like a typical bacterial cell, has no nucleus. So what we have is a nucleus-like. That is why they call it nucleoid, and it is not membrane bounded. So you need to know that. Then the function of a nucleoid that we see in bacteria, dividing bacterial cell, is that it is the site of genetical material. That is why the genetical material is being located. So then it is also the site that controls cell growth, reproduction, and cellular metabolism, through which there is going to be gene expression. This one is actually expected because if genetic material reside anywhere, that means it is going to be the site at which there is going to be initiation of gene expression. Then it is also a it also function because it is a compacted structure which allows for transcription and replication. Just like I told you, because it is the site that contains the genetic material, you will definitely expect that it is going to be the site at which transcription and replication is going to take place. It's going to take place. Then the physiology and mechanism by which nucleoids work is that the DNA, which is a supercoil, is attached to the cell membrane. Okay, so that is where it functions. Then it also, the protein also helps to fold and stabilize the DNA during transcription, as well as during their binary fission as well. So please, you need to take note of this. So, and this also perform a very significant role by allowing microorganisms, especially bacteria in this circumstance, to adapt, genetically adapt to any change or changes in the environment. So any bacterial cell that contains nucleoid, or will be able, as a result of a gene or genetic site of gene expression, being the site of transcription, being the site at which replication. So the only thing is that it is going to take them a very short period of time for them to be able to adapt to that environment. And that is very possible because they have nucleoid, which is the site of genetic material, the site of replication. It is also the site at which transcription will also take place. An example is found in Escherichia coli, which immediately alter the gene expression in order to rapidly metabolize different types of sugar. For example, the primary source of energy in E. coli should be glucose, but in the absence of glucose, in the absence of glycogen, in the absence of other primary sugars, E. coli has the capability to be able to make use or utilize that of so we will not forget lac operon, lactose. That is, whenever there is no lactose, the genetic expression of E. coli will just be such that it is going to adapt very, very quickly to that environment. And before you know it, the microorganism that will definitely make use of their primary source of carbohydrates or sugar will definitely be glucose. That Escherichia coli is going to start metabolizing any other substrates, any other sugar that can be found within the reach of that. So this Escherichia coli can only do that because of the presence of what is called nucleoid. Okay. Now the next thing is the cell wall. The next thing is the cell wall. Don't forget that cell wall is mainly made up of peptidoglycan, that is murein, which is a mesh of sugar. It's a mesh of sugar chains that contain NAM and NAC, N-acetylmuramic acid, as well as N-acetyl uh, glucosamine, which are cross-linked together. So this is where you have the peptidoglycan, which contains that. So the function is that they help the organism, they help the microorganism or the bacteria to be able to maintain their shell shape, as well as prevent osmotic lysis and help them to be able to resist mechanical stress. So this is also very possible because of this, and this is very useful. It's very useful in resisting antibiotics. So, for example, the beta lactams, it is the cell wall or the peptidoglycan that help the organism, the bacteria, to be able to metabolize, to be able to actually prevent beta lactams antibiotics such as penicillin. They contain that enzyme that will just break it down and make antibiotic and resist antibiotic for them to be able to function. So it also contains some certain enzymes such as autolysin and transpeptidase, which are used in building wall during cell division. During cell division. So, and just like we said, it also helps bacteria to be able to survive the harsh environment. An example is that of Streptococcus pneumonia, which resists drying in the lung. So as we can see it, we can see this dividing bacteria, this dividing bacteria cell, and this is the position of the cell wall, which contains NAG and NAM, which is made up of peptidoglycan. Glycan is a sugar, peptido, that is a peptide. So we will also look at the structure of this. But at this point in time, you will know that the classification of bacteria into gram positive and gram negative is basically because of the thickness of the cell wall. Okay, so that has been able to help us to be able to do that. Then it also help the bacteria, it help the organism to be able to interact with the host. So because it is located at the external, so it help the bacteria, this wall, because the outside, it is the external. So this is where communication, this is the structure. Cell wall is the structure by which this bacteria communicates with its host and the host environment. So, and it's very possible, it's very useful for the microorganism to be able to perform their role as a pathogen, as a disease causing organism, because it help them or protect them from drying up, and also help them to be able to interact directly with the immune system. So that is that. So the next thing we are going to look at is the capsule. And another word for capsule are called, I mean, it's called the glycocalyx. So you can see it in some other textbook, whereby they will also refer to capsule as the glycocalyx. So this is actually made up of polysaccharide and polypeptide. It's made up of polysaccharide as well as polypeptide, and it is a slimy. Yeah, it is a slimy of outer layer which consists of this. So that is a very, so it is the capsule that will make them to be able, that will protect the microorganism from phagocytosis. Don't forget that the immune system will definitely want to react, will definitely want to swallow up if pathogen is being sensed. So because of the presence of capsule, like pneumonia, because of this capsule that has been able to protect this organism from the harsh effect of the immune system. So because the more the phagocytotic complexes, like the macrophages, they are professional phagocyte, that is, they want to swallow them. They want to grip the pathogen and be able to lyse them. So because of the capsule, the presence of capsule in some of these microorganisms, it has made it very, very impossible for phagocytes to be able to even swallow them, so thereby protecting them. But don't forget that what do they contain? They contain polysaccharides as well as a polypeptide. So this is capsule also help them to be able to evade, okay, just like I said. It prevent phagocytosis. It help them in adhesion, that is, it help them to stick to the surface, and are very, very important in biofilm formation. So because they are slippery. So don't forget that when you just put a bucket full of water and you leave it standing for a lot of, I mean, let me say for like 48 hours. So, and you pour it away before you come back, you just see that when you pour it away, you just see that the bottom of that plastic is going to remain slimy. So the slimy is what is called the slimy component is what is called biofilm, and that has protected the microorganism from what being swallowed. Direct effect by phagocytes. So it is also an energy intensive because it is capsule. All capsulated organism would definitely use it for infection. For infection, it's going to help them to be more virulent. Don't forget the pneumococci, that is the theory that actually led into the discovery of DNA to be either to be located in this. So is as a result of the action of capsulated organism. They discovered that some capsulated organism were more virulent than the non-capsulated organism, because capsule will exert some additional virulent role on the organism or the bacteria that contains them. Okay, so it also help in colonization and immune evasion. As you can see it, because capsulated organism can stand for a very long time, can withstand high temperature, can withstand a lot of pressure. An example of that is a Klebsiella pneumonia, which thrives on medical devices. So medical devices, they are being if you want to sterilize medical devices, you subject them into autoclave condition, that is about 221 degrees centigrade. Okay, and any capsulated organism is capable of even withstanding high temperature as well as a high pressure. So that is what they do. So the next structure of a typical dividing bacterial cell is a ribosome. The structure of ribosome is such that the 70S ribosome consists, that what is found in a prokaryotic organism. We call them 70S, that is subvat. Okay, so 70. So which consists of 50S as well as 30S. So this is the large portion of the ribosome, which is this one. The upper one is called 50S. Then the lower one, which is the small, is called 30S. So which composed of ribosomal RNA and protein. So it is made up of that. You can see the structure over here, that it is, and it is the site at which protein is being manufactured. And just like I said, it synthesizes protein, which is used for growth as well as diffusion. Then it is also, ribosome is also a target for antibiotics. For example, tetracycline, chloramphenicol, and some other antibiotics. Their main target in pathogenic organism will always be to target the ribosome, because they discovered that once ribosome is being targeted, most of the vital enzymes of that bacteria will no longer be able to synthesize. So it will be very impossible for protein synthesis to carry on. And once there's no protein synthesis, that means there's not going to be any virulence and there won't be any diseases. So it supports rapid protein production in any nutrient-rich environment. An example is what happens in Mycobacterium tuberculosis, which grow very slowly due to their reduced activity. So that is very, very important. So because ribosome in this Mycobacterium tuberculosis is less developed, that is why they are capable of they cannot grow rapidly. So they are slow growers. They are slow growers. Okay. No, so the next structure we will be talking about is a cell membrane. Cell membrane. It is a bilipid layer which contains protein. So we can see it. This is the upper layer. This is the upper layer which is made up of phospholipid, and the lower layer is also made up of phospholipid, and you can see in between. But you can see this is a membrane protein, transmembrane protein, and this is another protein. So cell membrane exists as a lipid bilayer in which protein are being sandwiched. So no steroid except in mycoplasma. So cell membrane do not contain steroids as a lipid except what we have in the mycoplasma. And if you go back to the classification of bacteria, you will see why steroid in mycoplasma is also is used in naming it. So that is that. So the function of cell membrane is for control of nutrients, control of transportation and nutrient transport. It is the site of respiration and also the site of energy production. So if it is also involved in septum formation, which is during binary fission. It plays a very vital role in binary fission. So the cell membrane ecological role, that is, it helps bacteria to be able to modulate their fluidity for the environmental adaptation. That is, because of their cell membrane, it's very possible for microorganisms to be able to be flexible in any environment that they find themselves. An example is that of Allobacterium species, which still survive in high salt or high salinity. Okay, so the next structure is a plasmid. So plasmid is you can see, is circular in nature. It's like when you are winding, you wind double helix. So it's a small circular DNA which is capable of replicating independently of the chromosomal DNA. And just like I emphasized in my earlier video that wherever you see plasmid, any organism that possesses any bacteria that possesses plasmid, they are being there in order to be able to help them to be able to resist antibiotic. It is used for antibiotic resistance. Apart from that, that is what differentiates antibiotic sensitive strain from antibiotic resistant strain as well. So they play a very vital role also in the genetic composition of bacteria. So we will see this in more detailed structure. So it carries resistance. It is responsible for virulence and metabolic genes. So any microorganism or any bacteria that has plasmid, you will know that that plasmid, the structure is going to be there. It's going to be that bacteria is going to be more virulent than bacteria that do not have it. Then it is the main agent when you conjugation, that is the only thing, that is the only material that two bacteria that do not have plasmid, and the one that have plasmid, that is the only material that will bring them together through the process of conjugation. So that is the genetic material that is transmittable from one bacteria to another through a process that is called conjugation. So the ecological role of plasmid is that it increases their adaptability to the environment, to any environment, be it antibiotic environment, very inclement environment, and very uh, non and even very also help them in order to be able to adapt to uh, very harsh condition or harsh environment. So it is for their survivability. So it has the microorganism or bacteria that contain plasmid is going to help them, is going to contribute to their survivability. So example of that is E. coli, which contains R plasmid that help them to be able to resist antibiotics. So the next structure we are going to consider is a pili. That is the structure. It is very short. It is a protein. It has a protein-based projection, which is called pili. So you can see this is a pili, okay, and it contains a protein that help them to be able to, so we can see that it enable them to be able to attach to surface, to hard surfaces, and also help them to be able to exchange extra chromosomal materials or DNA via these pillars. So that is one of the function or functionality of pili. So any organism that contains pili, that is what they use it. They use it as for sexual exchange. For sexual exchange. Okay, so it initiates infection and also form what is called a biofilm. Example of that is what we see in Neisseria gonorrhea, which help this bacteria to be able to clinch, to attach to the host tissue. So, and lastly, we are going to consider the structure of flagellum or flagella. So, it is very long, longer than pili. It is capable of rotation, and the reason or the structure that give them that rotational ability is because of a flagellum. It provides motility and chemostasis. An example is cholerae, which help them to be able to swim and colonize intestine. Once again, I really want to appreciate you. In this video, we have been able to consider different types of structure that we can find in the structure of a bacterial cell, the component, the body plan. We consider that of cell wall, capsule, or what is called a glycocalyx. We look at the structure, the function, the physiology, the ecological role, case study. We also look at the ribosomes as well as a cell membrane, which is a plasma membrane. Plasmid, we also consider plasmid, fimbriae, or what is called pili. We look at flagella. We look at mesosomes as well as a nucleoid. Okay, so I want to really believe that I you must have gained one or two things in this. So I want to see you in the next video. Don't forget to subscribe to my channel, Omega Bio-Edutainment, as well as Bio-Nexus. Until next video, I want to thank you for your listening attention. Thank you.